Hello, welcome back to the Computational Genomics Lab. Today we're going to continue our adventure using genomic ranges and looking at features of the human genome. Last time we just learned how to sort of index them. Now we're going to have some fun with them and explore more about our genome. Okay, so to do that, we're going to go again to lessons 08. We are on the last one of this section, and then we move to the last hard level before your choose your own adventure guide. So you're almost there. All right, so let's get going here, where we're just going to look at like how big are genes, how many genes are there, how many genes of each type, what's the biggest gene. We're going to do some of that fun stuff, and while we're doing that, we're going to learn some redo some of our basic R skills and put it all sort of together and have some fun learning. Okay, so you can load your results. I have my GenCode GR length. That's the key thing you need right there. And let's make a data frame of just protein coding genes. So if we look back at GenCode GR length, we have this column called gene type. Oops, here we go. And there's protein coding in here. And those are mRNAs that get sent to the cytoplasm and translated by ribosomes into proteins. Okay, so let's make a table of just those in doing one of our indexing exercises in base R. We're going to go into that table and just grab out things that match protein coding in the column type equals gene. Okay, so we're doing a double filter here. So we're using the function filter. We're going to go into that table we just looked at, into the gene type column. It has to equal this, so no caps, no difference, exactly the way it's spelled. And then it also has to have type equals gene, because we could have exons in there as well. So we see here protein coding gene transcript, but we also see exons. We might just right now just want to look at just the mRNA gene names, for instance. Okay, let's do that. So that's what this will do, is by saying I want protein coding, and then in that next column saying I just want gene to match. So double filter. There we go. And if we look at our mRNA data frame, looks good. We got all the same information. So we just went in and grabbed all the columns for the things that matched gene, and then the things that matched transcript or gene ID, so not exon. So you can see how that can be a really useful way to dig into a table, is going into one column broadly, and then within that column another subcategory, and perhaps a third or fourth, depending. Okay, let's take a look at the widths of our mRNAs that we just selected. And first of all, we can also say there's about 21,859. Fun fact, that's how many genes there are in the human genome. Mouse genome, sorry, my bad for now. Human genome is also available as well for level 10. Okay, so we're going to make a histogram, log 10 space, of our mRNA data frame of width. So remember, we still have all these properties here, like width or length of that gene, how long is a gene, and we'll give it 60 breaks. Okay, and we can see it's doing its plotting. It shouldn't take that long. Let's see. Oh, figure margins are too large, so that means I have to respace my environment. So let's do this. And now rerun it. So if that happens to you, you can see, isn't that silly? It has to be bigger than some minimal thing. So I just usually stretch it more and more. Okay, so we can see that most of our genes are like around 10 to the 4th, so about 10,000 long, and we have some as much as 10 million. So let's make a same data frame and look at how long these link RNA genes are, and commonly now referred to as LNC-RNA. Happens to be my license plate, by the way. Okay, so we now have that data frame that's just long non-coding RNAs, and there's about 5,629 of those. Okay, so not as many as that 20,000 we saw for our mRNA data frame right here. Okay, let's look at the width of that. And we can see they do spread out the same length, but the mean is shifted more towards 10 to the 3.5, so a bit smaller. And you could do some interesting investigation there if you'd like to see why are so many genes this length, other features. But you can also take the mean. So let's take the mean of the long non-coding RNA gene. And it's 20,164. Let's do the same for mRNA, 49,000. So it's about half, somewhere around there. Okay, so long non-coding RNA genes are about half as long as mRNA genes. Fun to know. And we can look at all the different sort of data types that are in here. We can see there's 60,000 start codons, stop codons, just a ton of information. I'm going to skip that. And if you want to continue on here, I'm going to skip the lines above here and go right to 111. It's just more exploration of annotations and things like that. There is some annotations that are one letter long. How could that be? This walks you through that if you're interested. Otherwise, we can skip ahead here to 111 and use the promoters function. This thing's awesome. Okay, so we don't have promoters as an annotation in our genome annotation file because it's arbitrary. Typically, some people like 1 KB with 500 downstream, 500 upstream, or 1 KB upstream, 1,000 upstream, 500 downstream. It's somewhat settled, but you know, it's kind of arbitrary. And the best thing to do is just try a bunch of them and see if you go smaller, does the phenomena change, or go bigger, does it change, and then make interpretations that way by having all the different permutations. Okay, so let's do that. So we can go into our GenCode GR, our big one that has like everything in it, and there are GenCode GR type gene. We've seen that index before. We're doing a little different here. We're going into the type column instead of the @ element data like we did last time. And we're saying, give me upstream 0 and downstream 0. So we're retrieving the start, essentially, of all things that are a gene. So this would include link RNAs, mRNAs, all of those things. We didn't separate them in this case. And we're going to pipe it in the tidyverse pipe to data frame. We're calling this TSS for transcriptional start sites. So good use of the promoters function. We take a look at that. And what we can see is we have our sort of high-end level of all of this information for 55 things, which is more than mRNAs and link RNAs combined, so there must be other things in here. Whoa, excuse me. But yeah, there's just a ton of information that can be gleaned from here. I'm going to clean up while this reloads and look at our TSS data. And we also have the strand here. Remember how our ATACseq peaks, when we pull in those files, we did some of that testing before, they had the star, and therefore we don't know the strand. In this case, we have plus and minus strand. Determined for which way the gene's going on the plus strand, on the plus strand or the minus strand. I always think top left to right, bottom right to left. Okay. Excellent. So let's start. Where did we go to 111? I accidentally went back. Okay, so now we have a really cool information of where each gene starts in the genome, what chromosome it's on, and we can make a window around that to make it bigger or smaller. So let's look at summary. It's 55,000. It's the same as what we would see up here with 21 metadata columns. Okay. So we're going to make a data frame where we filter all genes here. So we're going to make it now back into a data frame, GenCode GR as .data frame, and then filter just all the genes as we did before. And now we have those 55,000 genes in a data frame. Same information here, except for we're really only going to be looking at things that are a gene, and then we can look at their widths, and we have the chromosome start and stop of the gene, as well as that gives us the TSS as well. Okay, there's a bunch of information in there. Okay, so we can also then arrange them like we could here. We could arrange them by width, so I can click on width, and the smallest is 10, and the biggest is, wow, 4,434,882. That's long, and we can look here. It's a protein coding gene called gallantail2L, okay, and some other information about it, but that's just, we can do that same function we just did there using the arrange function, so we can open that GenCode data frame we just made, and then sort it by the column of width, which we just did virtually. And now when I open this, it will automatically, oh, it didn't. Interesting. I don't know. Well, there's a way to GenCode DF. Oh, we ordered them here. Okay, so in the console, because we just piped it to a head, we listed out with this head command the smallest ones that that's the same when we did arrange. It went from smallest to biggest, and we did that manually with code this time instead of clicking on the buttons. So so if you want to take the top 1,000, you could do that instead of having to scroll down and all that kind of stuff. You say sort head to 1,000, print those out, and you have your top 1,000 smallest or biggest genes is what would be the common practice of that. Okay, so there they are in the console. So if we want to find the longest gene in the genome, we could switch it to minus width. I'm not going to do that so I don't have to change my screen, but it's ordered down there on your console. And if I look at GenCode DF, which we just did that to, width, it's going to now be the biggest gene, which will be this gallant2 that we just looked at, arranged that way. So just that you can also sort your tables using code, which becomes handy quite often. Okay, let's find the longest gene, max width filter. So we're going to make an object called longest gene. Instead of looking through these tables, we'll just make an object and go into GenCode DF, pipe it into a filter of width, which is the max width, and it knows width, so we didn't have to index it because we piped it using the tidyverse. That's why I love tidyverse. And then we can just simply click and look at longest gene, and it is gallant2L, as we saw when we sorted it before that. So, okay, let's say we want to find the longest exon. That's a little bit trickier because each gene has multiple exons. We have to think about how do we grab all the exons and do some summary and find out the longest one. So let's do that right here. So we'll say total exon length. We're going to go into GenCode GR type and equal exon. Last time we did gene. Here we're saying just give me all the exons. And then we're going to use genomic ranges reduce, which is going to do this here and take the maximum width of the overlap. So the furthest to the left and the furthest to the right of the two objects that have an overlap. So these are an example of exon 1, exon 2. This would be the reduction. This would be the intersection. Only the part that overlaps. Okay, so we're going to do that. So that's kind of cool. Like in that one reduce function, we've taken all the exons and said, okay, just give me all the exons and give me the longest possible overlap of a given exon, because each exon might have a different start and stop. There's a lot of them. Okay, so let's do that. Let's make a table first that just has our exons here, using that index into type. Okay. So total exon length here, it's really long list. And let's run that, oh, we summed up, yeah, the length of all the exons. Okay, wow. Okay, so the total exon length is, like, there's 130 million sum. Is what we just did, and we reduced it and then summed the total width of all exons. So now we can ask the question, what percentage of the entire genome do exons represent? And we divide that by 3.2 billion, and it's roughly 4%. So exons are only about 4% of the genome of all the genes, and we could do the same thing for genes as well. I think this is just making histogram. Let's look at the longest gene this way. Let's continue looking at our exons. I think this is a good exercise. So this is making exon lengths. Instead of last time we piped it right into reduce, we're doing the same thing essentially here. And now if we look at our exon lengths, it's in this vector thing, and it has element data here. But let's go back to our code. Okay, so let's take a histogram of this exon length we just made, again using that reduction thing described up above, if you want to go back and look at that some more. So let's do a histogram of the width of exon lengths. So now it's the reduction of each exon individually rather than the sum of all of them, which we did last time. Okay, so let's do that. Now we're going to get the widths of all of them. I think I'm going to make the x-axis limit a couple less zeros. Oops, too many. Okay. Add one more. Oh, come on. Okay, so I guess I did have it right. So if we zoom in here, we can see most exons are less than 1,000 nucleotides. That's actually quite interesting, I think, that they're this uniform. They don't have a wide distribution. So exons are like kind of, I mean, relatively uniform in length for very high level perspective. Okay, so the mean of the width, what's the mean exon width? We can get that here with average exon length is 454, so it was off by 2, but it's, yeah, so the average is about 454, and I bet the standard deviation isn't that much more. So let's look at the same thing where we reduce all genes into one length object, so to speak. So we're basically going to say, here's the if there's multiple gene bodies or different start sites, essentially, so if one gene starts here and one gene starts here, that might not be an exon problem, but we'd want to take the biggest gene body we can just to see how much of percentage of the genome gene bodies are, and not the exons like we did last time. So that might be an easier way to think about it. You have a big gene that starts here, and you have a smaller version of the gene that starts here. We're going to end up taking the bigger one. Okay, so that's the same reduce with sum. We're going to get the total length of genes in the genome and divide that by the genome length. Whoa, okay, 40%. Now we're talking. So genes take up a large portion of the genome, about half. That's cool. And this includes all the things called genes in here, those 55,000 things, combined make up a total of 40% of the genome. Okay, so I think that's interesting. I think somewhere in here I marked the human genome one. You can compare which one's longer. Human genome genes might be a bit bigger. And this is just doing the same thing for gene body widths. We're going to make a histogram. Oops, I didn't make this. And now I can get a histogram of the widths. And we can see roughly the same thing, except for around 10,000, and we can get that actual number. Let's see how close I was with, oh, here it is, 26,000, so it was under by half this time. Okay, that's it. So there you go. You learned, I hope, how to index into GRanges to find things you might be interested in, such as the width of a gene, which ones are mRNAs, which ones are link RNAs, or your favorite type of gene. And now we're going to move on to grabbing our ATACseq peaks and using these genome features to do overlaps. So we're going to find overlaps with our ATAC peaks into these genome features. So that's why we had 2 full classes on it. All right, let's go put it to an application in the next class. See you there.